Thank you for that introduction. I'm happy to be here today. This presentation reflects my views and should not be construed to represent FDA policy. In the next 20 minutes, I hope to provide information that will assist in the preparation of a well-organized sterility assurance application or building a better one. I intend to cover three main topics. First, I will discuss some best practices when preparing a submission for a sterile product. Then I will describe some common issues that lead to information requests and deficiencies issued by the product quality microbiology reviewers. Finally, I will briefly discuss some references that provide details about the type of sterility assurance information to include in an application and the organization of this information in an application. Using best practices when preparing an application can benefit many groups. First, the application holder benefits. When an application is well prepared and organized, there is the potential for less information requests and deficiencies, and possibly a faster completion of the review process. Second, the application product quality microbiology reviewer benefits. A well-prepared and organized application can be reviewed more efficiently. Third, patients benefit. A well-prepared and organized application with few to no issues can potentially be approved more quickly. Needed drug products can potentially reach the commercial market more quickly. Tip 1. One of the most important best practices that I can emphasize is the use of informative narrative summaries. All sections of Module 3 of an application that contain the product quality microbiology information can benefit from having well-written narrative summaries. Summaries are needed to describe the general programs at the facility and the manufacturing processes specific for the drug product. Diagrams and exhibit batch records cannot adequately tell the manufacturing story. Summaries should contain adequate details to explain what studies were performed, why they were performed, and how they were performed. Well-written summaries contain details consistent with the associated reports. The summary of a report should contain information that matches the details within the report. Well-written summaries provide rationale for the processes and the studies associated with the manufacture of a drug product. The reviewer should not have to guess or assume why a particular study was performed or is provided in the application. Tip number 2. Remember to include references to drug master files. If a specific sterility assurance information cannot be placed in the application because it is proprietary, then a reference to a DMF file, drug master file, for that information is needed. The reference to the specific DMF should be placed in the relevant sections of an application. This can be done in multiple places. A current letter of authorization from the DMF holder should be placed in Module 1 of the Electronic Common Technical Document. This LOA should contain sufficient details for the location of the sterility assurance information in the DMF. A reviewer should quickly and easily find the relevant information to review in the DMF. Details in the LOA should include the date of the specific DMF submission where the information is located and section numbers or page numbers containing the specific information. Now that I have described some best practices, I would like to focus on some common issues that can lead to information requests for information or deficiencies. One of the most common causes for requesting additional information is conflicting information identified within the submission. Conflicting information is found between narratives in different modules, between narratives in different sections, and between summaries of documents and the details in the document being summarized. These documents can be validation or qualification reports, standard operating procedures, or diagrams. For example, A reviewer reads in section P33 of an application that line 4 and filling machine Z are proposed for the commercial filling process of the drug. However, in section P35, the reviewer reads in the process simulation report that the media fill was performed using filling line 10 and filling machine X. Which section does the reviewer believe to be accurate? Is there a typographical error in the report or in the summary? Was the wrong report provided? Or maybe the applicant intends to use both lines, but they forgot to indicate this fact and they forgot to provide the second report. The reviewer must request clarification. Another common issue: absence of rationale or justification for a study. Validation studies are necessary to demonstrate that commercial production processes will be performed as described. However, validation studies may not be identical to the intended commercial production process. Therefore, it is important to provide an explanation of how the validation studies represent or support the commercial production processes. And it is important to provide a justification for an alternative validation scheme. Let me give you an example. In sections P33 and P37, the applicant indicates that the 10 ml vial is the container of the drug product. However, in section P35, a validation report is provided that contains data for depyrogenation studies using 2 ml vials. No explanation or rationale is provided for how the depyrogenation validation study with 2 ml vials is related to commercial depyrogenation of the 10 ml vials. Does the reviewer assume the applicant submitted the study in error? Does the reviewer assume the 2 ml vial is the worst case vial? The reviewer must request clarification. Another common issue: no information about components or equipment received as sterile or depyrogenated. Some items are received as sterile or depyrogenated or both. Examples include product stoppers, vials, filters, and single-use bags. These items may be purchased already sterile or depyrogenated, or these items may be purchased and sent for additional processing. In both cases, another entity is performing a manufacturing process for the applicant. The applicant, however, is responsible for describing in the application the items that are affected by this and the specific processes. They also are responsible for identifying the entity performing the process and directing the reviewer to the location of the complete validation information. This information should be included in the application. However, if the information is proprietary and cannot be placed in the application, then it should be placed in a DMF, and the applicant must provide the DMF reference and a letter of authorization. Another common issue: the sterilizing filter for the drug product is not identified in the autoclave loads. There could be different types of filters used during the manufacture of a sterile product. These filters are often sterilized in an autoclave by moist heat as part of an equipment load. These filters can be air filters, bioburden reduction filters, and the product sterilizing filter. The product quality microbiology reviewer is interested in the sterilizing filter for the bulk product and needs to review the studies that validate the sterilization of the product filter. Using only the word filter to describe an item present in an equipment load does not provide enough detail about the type of filter and its role in the manufacturing process. The reviewer cannot assume that an item called filter in an equipment load is the drug product sterilizing filter. Filters in equipment loads should be identified with sufficient details to determine their function. Let me give you an example. A validation study is provided for the sterilization of equipment load 4. The load contents are described as filter, filters housings, scoops, manifolds, and bowls. The word filter and filter housings do not provide enough information about these items. Because other types of filters are used in the manufacturing process, the reviewer cannot have 100% certainty that these words refer to the product sterilizing filter and whether the validation report for this study is even relevant. A reviewer must request clarification if load number 4 contains a product sterilizing filter. Another common issue. Bioburden monitoring of the bulk solution is not described. Regardless of the manufacturing process, the bulk drug solution should be monitored for bioburden. There are 3 different common deficiencies associated with bioburden monitoring. First, there is no description that routine monitoring of the bulk solution will occur during commercial production. Second, bioburden monitoring may be described, but the point of monitoring during the manufacturing process is not described. Finally, bioburden monitoring is described, but the location of monitoring in the manufacturing process is not adequate. This is one of the more frequent points of clarification. I'd like to elaborate on this. We recommend that bioburden monitoring of the bulk solution occur before any filtration step. Bioburden monitoring is an important in-process control that can help one understand the microbiological quality of the bulk solution. Excessive microorganisms in a bulk solution can overwhelm a filter. Additionally, the presence of excessive microorganisms can result in accumulation of cell debris, toxins, and metabolites. These items most likely will not be retained by the filter, and they can enter the drug product during the filling process, where they can affect product quality and create a patient safety issue. This is an example of a very general manufacturing process. It begins with a compounding step, followed by a holding period of the non-sterile bulk solution. Then there is a first filtration, followed by another holding period, and then the product sterilizing filtration step at the second filter, and finally filling into a container. We recommend that monitoring of the bulk solution for bioburden occur after compounding and immediately before the first filtration. This is shown by the purple arrow. Monitoring immediately before filter 1 provides the most accurate information regarding the level of microorganisms that may have experienced growth during the hold of the non-sterile bulk. If one samples the bulk solution after filter 1 but before filter 2, the sterilizing filter, the true bioburden level is unknown, and there is a risk that the excessive microbial metabolites and toxins have passed through the first and second filters into the product. And I will say that alternative monitoring plans should be adequately justified. Another common issue: no pressure and vacuum conditions are used for container closure integrity. The microbial and dye ingress tests for container closure integrity rely on the dye or the challenge organism in the emergent solution reaching the interior of the container in order to be detected. When the dye or challenge organisms encounter a breach in the container or closure, they must pass through the tortuous path of the breach to reach the interior of the container. Use of pressure and vacuum helps remove air bubbles, particulates, and dried product from existing breaches. And the use of pressure and vacuum ensures that a fluid pathway is established through a breach to allow ingress. We consider using both conditions as a best practice for complete removal of any blockage of the leak path and for creation of a fluid pathway. However, we are flexible on these conditions, and alternative test conditions should be adequately justified. Another common issue: unacceptable incubation conditions for biological indicators. Another concern is the use of biological indicators with reduced incubation periods. For Geobacillus stereothermophilus used in moist heat sterilization, the incubation period is typically 7 days at 55 to 60 degrees Celsius. However, commercial BIs of this organism are available with 24-48 incubation periods. although the certificate of analysis for these BIs may indicate that the manufacturer prepared them per an FDA guidance. The FDA guidance that is referenced and describes the preparation of these BIs is a guidance that is intended for BIs used in healthcare facilities and not in the manufacturing setting. We are concerned that the 24 to 48-hour incubation period may be too short to allow growth and detection of low numbers of sublethally injured spores. Therefore, we recommend the use of a 7-day incubation period for G. sterothermophilus for moist heat sterilization studies. Another common issue. Media fills are not representative of the maximum production conditions. The parameters and conditions of media fills should be representative of maximum commercial aseptic filling conditions. Now, what does it mean to be representative? It means that the media fills are designed to simulate all aspects of commercial production. The container closure system used in media fills should be similar to that of commercial production. The media fill durations should approximate the maximum commercial filling process. Routine and non-routine interventions that are performed during commercial production should also be simulated during the media fill. Environmental monitoring and growth promotion studies are performed during media fills, and units that may be rejected and discarded during certain activities in production are treated similarly during the media fill, and vice versa, and the criteria for these actions are described. A rationale is needed for media fill conditions that do not reflect commercial production filling conditions. We understand that many applicants must qualify different products, container closure systems, production conditions on one filling line in many cases. However, if explanations and justifications are not provided, then the reviewer must request clarification. Another common issue, incorrect use of pooling for endotoxins testing. Pooling of samples prior to endotoxins testing is allowed for small volume parenteral products. This means volumes of 100 mL or less. However, if pooling is performed, the FDA recommends pooling of a maximum of 3 units. Additionally, the maximum valid dilution must be adjusted proportionally. You must divide the MVD, the maximum valid dilution, by the maximum number of pooled units. Our concern with pooling is that it can potentially mask high levels of endotoxins present in one unit by the dilution effect when that unit is combined with other units with low levels of endotoxin. The MVD must be decreased so that a more concentrated solution is tested. Another common issue, incorrect endotoxin limit for product release. Monographs for some drug products in the USP may list an endotoxin limit. However, limits in the monographs may not be correct because they do not reflect the current labeling that defines the maximum patient dose. The acceptable level of endotoxins exposure for an injectable drug is based on the total amount of endotoxins in the maximum drug dose received by a patient in a 1-hour period. Therefore, the endotoxin limit for a finished drug product should be established based on the maximum patient dose and route of administration stated in the package insert that accompanies all products. It is essential that you perform calculations to determine the allowable endotoxin specification for your product and use USP 85 as a guide. My next 2 slides contain some helpful references with regard to organizing an application. The 1994 guidance outlines the information and data needed in an application for a sterile product. The 2004 guidance provides recommendations for the performance of aseptic manufacturing processes. The question-based review documents are white papers that contain specific details about information and data required for the common technical document submissions with sterility assurance information. There are 3 QBR documents that are helpful. QBR for sterility assurance of aseptically processed products, quality overall summary outline for terminally sterilized products, and the QBR for sterility assurance of terminally sterilized products, frequently asked questions. This last document, the frequently asked questions for terminally sterilized products, defines almost all of the product quality microbiology reviewer information that is evaluated for a terminally sterilized product. And many sterility assurance topics also apply to aseptically processed products. Although the documents refer to ANDAs, the information also applies for the preparation of new drug applications. This is a link to a web page that describes submission requirements for ANDAs. When you reach this page, you will find additional links to the 3 documents listed above. I hope you've gained some helpful knowledge about how to prepare the sterility assurance portion of an application. My call to action for everyone is to remember the following. Summarize sterility assurance information clearly and consistently. Provide validation data in the submission or give a reference to the specific location in a DMF. Review the common issues discussed in this presentation and use the guidances and question-based review documents listed on slides 18 and 19 as your guides for building a better sterility assurance application. I now would like to present 2 challenge questions. After I read the question, please enter your answers in the Q&A pod. The first is a true-false challenge question. True or false, if an application has an associated DMF, then the application should contain both a letter of authorization and from the DMF holder and a reference to the DMF in the relevant sections of the application. The answer is true. Whenever sterility assurance information related to the manufacture of a drug product is placed in a DMF, an LOA for the DMF should be placed in the application to allow the product quality microbiology reviewer to access DMF information. Additionally, a reference to a DMF should be placed at the specific locations in the application to alert the reviewer that the specific validation information is not within the application, but within a DMF. Second challenge question, fill in the blank question. The FDA recommends that pooling of endotoxins testing use no more than blank total units. Is the answer A, 2 units, B, 3 units, C, 4 units, or D, no pooling of units is allowed? The answer is B, 3. According to the 2012 FDA Guidance for Industry, Pyrogens and Endotoxins Testing, Questions and Answers, no more than 3 units should be pooled for endotoxins testing. Additionally, the maximum valid dilution should be reduced by 3, the number of units pooled. This concludes my presentation. Thank you for your time. I will be back for the question and answer panel. Please submit any questions you have for me to the question and answer pod.